can change, use also this pointer. This is for next slide. This is for previous slide.
Okay. And is there a pointer on that?
Yes, sir. This can be projected.
Whoop.
This is for the next slide. This is for the next slide.
Okay. That's okay. Let's go back.
And this is the pointer.
This is the pointer. Okay. Well, thank you very much. It's indeed a great honor, a privilege to talk at this meeting, which I think is a wonderful, wonderful effort. It's my first trip to India, and I must say it's an overwhelming experience to be invited to give this talk today. So thank you very, very much for inviting me. Now, what I'd like to talk about today is our work over many years on skeletal function and dysfunction, and I've called it a tale of molecules, mice, and men, and you'll see why hopefully in a moment. Yeah. Oh boy. I did something.
Sir, it connects. This one.
Oh,
This
that-
button is connected.
All right. Thank you. Okay, so I thought I would start off with basic principles so that everyone is on the same page about what we're talking about. And we'll start off with the composition of bone. This is especially long bones. And we know that long bones consist of cortical and trabecular components, an outer shell of cortical bone, which makes up about 75 to 80% of the skeletal mass. It defines the shape of the bone. And inside the bone, there's trabecular or cancellous bone, which is a network of connecting plates and rods, and it comprises about 20 to 25% of the remaining part of the bone. At a cellular level, there are, of course, cells and matrix. The matrix is an inorganic matrix with calcium and phosphorus being the major components in the form of hydroxyapatite crystals. And then there's about 1/3 is organic matrix, which is mostly proteins, type 1 collagen, and some non-collagenous proteins. And there are also some mucopolysaccharides. Whoop. I'm definitely challenged on this one. Okay. Now, bone consists, the cells of bone undergo a process called bone remodeling, or carry out a process called bone remodeling. And the bone remodeling, in the initial phase, there's a quiescent phase of bone where stromal cells, mesenchymal stromal cells, and bone lining cells cover the surface of the bone. At the initiation of the bone remodeling cycle or bone turnover, pre-osteoclasts, which are monocyte macrophage origin, will migrate in. They'll be activated to multinucleated osteoclasts to undergo, which will then resorb the bone. The osteoclasts will undergo apoptosis, and the process is reversed with osteoblast precursors migrating into the resorption area and being differentiated into osteoblasts. The osteoblasts lay down new bone, which is called osteoid, and the osteoid can be then mineralized to new bone and then eventually become more mature bone. Now, this process is modulated along with various other things by hormones, and I'm going to talk mostly today about parathyroid hormone and vitamin D. The FGF23, fibroblast growth factor 23, is another mediator which Dr. Karapoulos will talk about following my talk, I believe. So when the blood calcium is reduced, when the serum calcium goes down, there's a signal through the calcium sensing receptor within the parathyroid glands in the neck. And the parathyroid glands will release parathyroid hormone. This will then act on bone to resorb bone, to remove calcium and phosphorus, and to help restore a normal calcium. In addition, parathyroid hormone itself will reabsorb calcium from the kidney, and importantly, parathyroid hormone will stimulate the precursor of the active form of vitamin D, which is 25-hydroxyvitamin D, to be converted to the active form 125-dihydroxyvitamin D. 125-dihydroxyvitamin D will then enhance absorption of calcium from the gut, and it will also resorb bone when it's present in high concentrations, release calcium. The net effect of these actions on bone, on the gut, and on kidney is to restore a normal serum calcium and turn off the signal to the calcium sensing receptor so that no further increase in PTH, parathyroid hormone, occurs. Now, many years ago, we began to study parathyroid hormone. The major protein form of parathyroid hormone released from the parathyroid glands is an 84 amino acid straight chain peptide, parathyroid hormone 1 to 84, PTH 1 to 84. The entire biological activity, however, resides within the amino terminal 34 residues. PTH 1 to 34 can be synthesized and has all of the biological activity of PTH 1 to 84. And PTH 1 to 34 or PTH 1 to 84 can interact with a G protein linked receptor in the cell membrane, which is a 7-transmembrane receptor. And this will set off further downstream signaling. If you remove just the first two residues of PTH 1 to 34, PTH 1 to 84, it's inactive. Now, because PTH resorbs bone, we thought that almost certainly PTH would bind to osteoclasts, which are the bone resorbing cells. However, when we did a study many years ago with I-125 labeled PTH 1 to 34, we found that it bound specifically to osteoclasts, which are shown by the blue arrows, which are to osteoblast precursors, which are shown by the blue arrows, and not to osteoclasts, which are shown by the red arrows. It also bound to mature osteoblasts, shown by the double arrows, so that it bound essentially to specifically to cells of the osteoblast lineage rather than to osteoclasts. So then the question arose, is there evidence for a physiologic anabolic role for parathyroid hormone, because it does act with osteoblasts, which is unmasked by eliminating PTH? And so the approach we used was to ablate the gene encoding parathyroid hormone. It was a gene knockout study. And we asked, what is the phenotype produced by targeted ablation of the gene encoding parathyroid hormone? When we did this, we looked at 2 weeks of age and at 4 months of age, and we looked at the PTH knockouts, PTH minus minus, which are the PTH gene knockouts, compared to the wild type. And as you can see in the red boxes, there's much less of this black material in the PTH knockouts compared to the wild type animals, and that means that there's much less trabecular bone. At 4 months of age, you see the same thing in trabecular bone, but on the right-hand panel, but you also see an increase in cortical bone. So that cortical bone volume seems to increase in the PTH knockouts. You need parathyroid hormone to resorb the cortical bone. You need parathyroid hormone to make the trabecular bone. So why PTH is required for trabecular bone formation of fetus and neonate, and PTH participates more in skeletal resorption of cortical bone as the environmental calcium sources change in the growing animal. And we know that the mother, and through lactation or in utero, provides calcium requirements for the fetus, but in the older animal, the older animal has to fend for itself to get calcium into the bloodstream. And so what we did show then was that PTH 1 to 34 can be anabolic, PTH 1 to 84 can be anabolic, But if you just make the synthetic amino terminal molecule, this will be active. And so, long story, but it ended up being a very useful agent as an anabolic, as a bone-forming agent for the treatment of a disease called osteoporosis, which is a very prevalent disease, especially in older, individually in older women. Now, a genetic relative of parathyroid hormone, or PTH, is PTHrP. And PTHrP was discovered as a mediator of hypercalcemia associated with malignancy. And the malignant cell can produce PTHrP even when it hasn't invaded bone. The PTHrP can get through the bloodstream. It can act on bone to resorb bone, and it can release calcium and cause hypercalcemia. But the malignant cell, even when it's in the bone, can release PTHrP, and this will resorb bone, and this is called osteolytic metastasis, and this can also cause an increase in the blood calcium or hypercalcemia. Now, PTHrP end with parathyroid hormone of PTH and of PTHrP, which are similar. The rest of the molecules are actually quite different. PTH 1 to 84 is the major form of parathyroid hormone, and PTHrP comes in 3 isoforms of 173, 141, and 139 residues. But the homologous end is intriguing, and what we did to understand the physiology is to do a knockout of PTHrP, compare it to a PTH knockout, and then combined knockout, and compare this to the wild-type animals. And when you do this, you see that PTHrP, the animals are very small, and they have a reduced amount of bone. And this is true in the PTHrP versus the wild-type, and to some extent the PTH knockouts, and it's also true in the double knockouts. So what's